Hello. We will start in 5-10 minutes. Okay, let's wait for something and then we will start. Okay, so hello and welcome everyone to today's live class session on the course of Fundamentals of Protein Chemistry, which is being taken by Professor Shagata Dasgupta of IIT Kharagpur. And I am Srijan Sen, and today we'll be taking your week 9 live session. And so firstly I would be discussing with you the course in brief details, and then we would be discussing a few questions that will help you in solving the assignment and taking up further exams, future exams that you will be having. Okay, so I am sharing my slides with you all. So I guess my screen is visible to you all and I am audible enough. Kindly confirm. Any one of you can kindly confirm if. Okay, that's good. Fine. Okay. So in today's class, we would be mostly focusing on the cellular membranes and mostly talking about the membrane proteins and different functions of the membrane proteins and different transport mechanisms and all. So let us see. So if we look into membrane proteins, what we see over here, this is your phospholipid PLP bilayer, protein lipid protein bilayer, and this is mostly your cell membrane. And over here you have different proteins which have different functions to play during the transport of different ions and small molecules inside or outside of your outside the cellular environment. Okay, so there are different proteins. Mostly you have over here the glycolipids, then you have different oligosaccharides, followed by these are your integral proteins that are mostly inbuilt or inserted inside the cellular membrane. Then you have different what you call is the hydrophobic alpha helices, which are also a portion of different integral proteins. This portion is, as you know, is the phospholipid, and there are different small portions which you can call as the cholesterol. And peripheral proteins are present on the surface of your PLP bilayer, and then there are different lipid linked proteins that are mostly linked to the different glycolipids or the different phospholipids. So if you look into the membrane design, so it's the fluid mosaic model of the plasma membrane structure, where you have, this is the outside of the cell, whereas this is the inside where you have the cytoplasm and all. Over here you have the hydrophilic heads that are mostly with exposure to the water molecules, and over here you have the glycoproteins, or you can call the glycolipids and all. And to the inside of the portion you have the hydrophobic regions, and within this layer of this PLP bilayer you have the peripheral proteins, which are mostly attached to the outer surface of your cellular membrane, and integral proteins which are totally inbuilt, that is, it is present inside the sandwich structure, and it is enclosed in the with both the layers. Okay, and there are different other proteins that have different functions. So mostly these integral proteins are play roles in the case of channels, or as different ion channels, or as transporters for transporting small molecules or different small ions inside and outside your cellular environment or outside the cellular environment. Okay, so this fluid mosaic model is basically your fluid because it is pliable and it can be easily moved, whereas it's the mosaic model because it is made up of different kinds of molecules, like you have the lipids, you have proteins, you have different alpha helical structures which are also proteins, and then you have carbohydrates, cholesterols and all. So this is made up of different kinds of molecules. That's why we call it the mosaic model, and fluid because it is easily moved or it's pliable. Now, as we know that the cell membrane is mostly selectively permeable because it restricts based on size and on polarity. So if you see over here, we have the gases like CO2 and oxygen that can easily pass through. You have hydrophobic molecules like benzene, then you have small polar molecules like water and ethanol that can pass through the cellular membrane via different kinds of proteins and all, whereas there are large polar molecules or different charged molecules that cannot just simply pass through this particular cellular membrane. So the cellular membrane rejects or accepts molecules based on its size as well as based on its polarity. Now, the cellular membrane only lets certain substances in and out of the cell. So these are the different which I mentioned over here. So not all molecules can pass through. It looks at the molecule, it finds whether it is eligible to go in or can be thrown out, and then it lets in that particular molecule. Now this phospholipid bilayer. So what is this phospholipid bilayer? So here we have the water molecules, and on the inside you also have the water molecules. So the hydrophilic or the polar head, which is your water loving portion, is on the outer side, and then you have the hydrophobic tails, which are mostly non-polar portions, tries to be, or the hydrophobic portions tries to be inside, and it forms a sandwich like structure. So these polar heads are hydrophilic, that are water loving, whereas the tails are mostly your fatty acids, which are hydrophobic and does not love water. So it loves to stay inside the core of this cellular membrane and form that bilayer, phospholipid bilayer structure. Now there are some other molecules which you also, for which falls in the category of sterols, which is your cholesterol. So these are type of your steroids or the lipids. So what is the role of this cholesterol? It basically changes the fluidity of the cellular membranes. Like higher temperature stiffens the membrane, and lower temperature prevents membrane from freezing. Okay, so these are basically present in between the different cellular membranes, and mostly in these junctions, and this actually is helping in modulating the fluidity, the fluid kinetics of the cellular membrane. So what it does, at higher temperature it stiffens the membrane, whereas at lower temperature it prevents the membrane from freezing. There are also other proteins, basically your recognition proteins, where you have the proteins that have carbohydrate chains attached, like you have the carbohydrate groups present over here, and then you have the carbohydrate chains aid in the cell identification, that is, it acts as a signaling molecule, or it acts as for understanding the tag. So it acts as different tags. For example, you have the red blood cells that identifies viruses as foreigns. So these act as the different tags, and these have different roles in identification. And the next category of proteins is your receptor proteins, which is important for this is also a signal capturing molecule, like it carries on with a different kind of a signal downstream. Okay, so these are the receptor proteins. So it receives something. So what does it receive? What it receives is the ligand. So and then it can undergo different signaling pathways. Okay, so the proteins that receive chemical signals from other cells, such as your nervous system cells. So after it receives a signal, it starts functioning, and then like you have the feedback inhibition or the feedback activation mechanisms, and these have different roles to play. So this is the role of basically your receptor protein. So it receives the ligands, and depending on what signal it has to convey to the inner side of the cell or downstream signaling pathways, it conveys. Okay, then you have the channel proteins. Okay, so these channel proteins are basically involved in letting certain substances inside or outside the cell. So So these proteins lets in certain say you ions, or there are different kinds of small molecules, sometimes divalent metal ions and all. So these actually lets allows the entry or the rejection of different molecules or ions and all. For example, you have the aquaporins that lets in and out water. Okay, so this is one example of your channel proteins. Otherwise, you have the calcium protein, calcium channel proteins, and magnesium channel protein. There are a lot of channel proteins, and you can look through them. Now, two important category of proteins are basically your integral proteins that cross both the layers of the phospholipid bilayer, and the other one is your peripheral proteins that are only on the top half or the bottom half of the phospholipid bilayer. Okay, so basically these are your integral proteins. The red ones. The red ones are basically your integral proteins that pass through both the layers of the phospholipid bilayer, and the peripheral proteins that are only one on top of the phospholipid bilayer. So they are attached to the phospho. You can tell the hydrophilic portion of the cellular membrane. So what is the function of your membrane proteins, or why are they so much important? And research on membrane proteins is a very hot topic in today's generation, and it's going to improve day by day. So membrane proteins are basically very important and in understanding as well as in different research fields in the scientific areas. Okay, and even purification or expression of these membrane proteins are very difficult. Anyway, so what is the function of these membrane proteins? So they help in the attachment to the cytoskeletons and the extracellular matrix. They have different messages to convey via the different downstream signaling pathways. Then they have often they perform different enzymatic activities, and all we know that they are semi-permeable, so they help in transport, and they help in cellular recognition by different receptor ligand interactions, and by different receptor proteins and all, and different roles in the intercellular joining, like you have the caterins and the nectins, the caterin-nectin interaction, and that you can actually understand from the membrane proteins, and studying these membrane proteins becomes very important. So these are the few functions of different membrane proteins. So okay, so we studied about the integral proteins. So how many types of integral proteins are basically over here? So there is type 1, where you have the single transmembrane span or the N terminus in the ectodomain, whereas the C terminus is in the cytosol portion. So you can see from here. So all the 6 types are given over here, and this is the description, and from here you can actually understand the types of the integral proteins. So type 1 is the single transmembrane span, where you have the N terminus to the ectodomain and the C terminus to the in the cytosol. So then you have type 2, where you also have a single span where the C terminus is in the ectodomain and the N terminus in the cytosol. So the inside is the cytosol and the outside is the ectodomain. Then you have type 3, where you have multiple spans. So you can see that there is no distinction of the N terminus or the C terminus. It can, the C and the N terminus can both be on the same side or on the different side, that does not matter, but they are passing through the entire of the lipid bilayer. Now you have the type 4, where you have several different polypeptides assembled to form a channel. So you can have so type 1 and type 2 actually join together to give you the type 4. So they can be different types of polypeptides forming a channel. Then you have type 5, which is the lipid linked protein, and you have the type 6, where the proteins with both the transdomain component and the lipid anchor. So you have the lipid anchor and you have the transdomain portion. Okay, so these are the different types of the integral proteins. So what are the properties of these integral proteins? So because of their hydrophobic transmembrane domains, the integral membrane proteins are very difficult to isolate them in the soluble form, because with the cellular membrane they have the hydrophobic portion that does not actually solubilize your membrane proteins, and because of that, getting this membrane proteins in the soluble form becomes very, very critical, and at the same time it's a very challenging task to do. So for that you need different kinds of detergents, like you have the ionic detergents, then you have some charged or uncharged detergents that actually play different roles in bringing your membrane proteins to the soluble fraction, okay, or in increasing the solubility of these proteins. So the use of mild non-ionic detergents for solubilizing, purifying and reconstituting functional membrane protein systems. Okay, so here you have the SDS or the Triton X100, which is basically used for the purification of your membrane protein. So here you have the membrane protein, you have the non-ionic detergent, and it gets attached to your protein and it comes out of the cell membrane, and then you can bring it to the soluble fraction. So membrane proteins, as I was mentioning, is very difficult to isolate for various reasons. So a few of them include that it's very hard to overexpress large quantity of functional proteins. It's difficult to separate the proteins from the lipid membrane lipids, and then to get the protein into the solution for purification, it needs to be solubilized with a detergent, which might also destroy your protein, because high amount of detergent is going to affect your protein in a very problematic manner. Like when you are running an SDS page, what happens? You are giving a uniform negative charge to your protein, and that is why it is running towards the anode. Okay, so what happens when you give you give a high amount of ionic detergents or uncharged detergents anyway. So what happens is that it just unfolds your protein, and when the protein gets unfolded, it does not in any way come to the soluble fraction. So in one way, from bringing from insoluble to soluble, ultimately it remains insoluble. So this is another very major problem. So and it's very difficult to crystallize these membrane proteins with the detergent around the molecule. So different structural studies using X-ray crystallography, or you can say your NMR structure, NMR becomes a very, very challenging task. Okay, so how can you purify these membrane proteins? So membrane proteins already cover more than 50% of the current drug targets and about 30% of the typical cells protein. So solubilization of the membrane proteins. So solubilization of these membrane proteins can be accomplished by using some amphiphilic detergents that have both hydrophilic as well as hydrophobic heads. But we also need to keep in mind that the preservation of their biological or the functional activities is also that we need to keep in our mind. Like we cannot use such a detergent that is going to unfold your protein or going to destroy the protein's enzymatic or functional activities, and it should be also susceptible to denaturation during the isolation process. So what is the feature of detergents that you are going to basically use for your membrane protein purification? So detergents are amphipathic in nature and contain a polar group at one end and long hydrophobic carbon chain at the other end. So the polar group forms the hydrogen bonds with the water molecules, and the hydrophobic chain aggregates via different hydrophobic interactions. So these are known informations to us, and this is the same story as that we studied when we studied about proteins and alpha helices, beta sheets, about how proteins fold, and also this is the same story. And what happens at low concentration, the detergent molecules exist as monomers. Now when the detergent monomer concentration increases above a critical concentration or above a threshold value, the detergent molecules self-associate to form aggregates that we know by the name of micelles. Okay, now what happens that this is your critical micelle concentration or CMC, that becomes a very important parameter for selecting a particular appropriate detergent. Now this effective micelle concentration is very, very important because it is affected by other components like your say lipids, the number of lipids, the concentration of your proteins, how many number of proteins are there, the type of proteins that are present, your ionic strength, and the temperature, pH. So those will actually affect your micelle formation, or at what concentration is that micelle going to form. Okay, so that becomes a very, these are the factors that affects this CMC formation. Okay, so now what is this aggregation number? So this aggregation number is the number of monomeric detergent molecules contained in a single micelle. So the number of monomer units that is forming the single micelle, that is your aggregation number. Okay, so these membrane proteins, as we had studied, that these have different roles in transport kinetics or transport mechanism. So basically, we know that transport can be of two types. One is your active transport, and the other one is your passive transport. So passive transport do not require the need of any ATP molecules, so it does not require, you can say, energy to carry out the transport function. Whereas on the other hand, if you see your active transport, that is an energy consuming transport. Okay, so that requires ATP molecules to carry out different functions. So in the category of active transport, you have so in the category of energy consuming transport, you basically have the active transport and your exocytosis and endocytosis. I will be mostly focusing on the active transport. And on the other hand, where you have the no energy consuming transport, that is a passive transport. In that category, you have simple diffusion, then facilitated diffusion, osmosis, and often you have heard of the term tonicity that happens in the plant cells. Okay, so these are a few categories that falls under the passive and the active transport. So these you had studied even in class 9, 10, or even 7, 8. So this knowledge you already have, just a brief idea about all of them. So diffusion, so you can see a drop of dye which is placed in a water molecule, and this dye molecules diffuse into the water molecules diffuse into the dye. So it is a mechanism that is occurring, and both the dye molecules and water molecules become evenly dispersed. So what is occurring over here? The movement of molecules in a solution from a high concentration of that molecule in a direction of a low concentration of that molecule until equilibrium is reached. So that is your entire phenomenon of diffusion. Now here you can see diffusion across the membrane, where you have the diffusion between the lipid soluble molecules and different gases like oxygen and CO2, that is going from the extracellular fluid towards the cytosol, or and vice versa. Okay, so this is the diffusion process that occurring across a membrane. Now, why does passive diffusion through a lipid layer only occur with gases and small lipid soluble molecules? So if you see that these substances are, so this is a question, so these substances are non-polar and so is the lipid layer, these substances are polar and so is the lipid layer, these substances are non-polar, the lipid layer is polar, these substances are polar and the lipid layer is non-polar. So if you see, why does passive transport through the lipid layer only occur with gases and small lipid molecules? So if you see that the second answer is correct, where you have these substances are polar and so is the lipid layer. Okay, now coming to facilitated diffusion. So if you see that proteins form a hydrophilic channel which can allow the passage of different ions, like here you can see the chloride ions, and then water molecules are moving out or in to the cytosol or the extracellular matrix. And then, so why are channel proteins necessary to move hydrophilic ions through the membrane? So the correct answer would be ions are charged, they cannot go through a non-polar lipid layer. And because of that, you need, because you can see over here that these are the hydrophobic molecules, and for the ions to pass, it becomes very challenging for them to pass. So the ions are charged and they cannot go through a non-polar because the hydrophobic nature of your fatty acid tails, and for that you need these channel proteins or to allow the transport of different small molecules and ions. Now you have the passive transport by the facilitated diffusion, where you have a carrier protein that has a particular binding site for a specific molecule. Now when it captures that particular molecule, the gate opens and the molecule enters the binding site. Now when it enters the binding site, it also passes a signal to the inside of the cell, and then the inner portion of the your protein opens up and the carrier protein changes shape, thereby transporting molecules across the membrane. Okay, and then what happens is that after that molecule has passed into the cytosol or the inside of the membrane, the gate again gets closed, and in this manner your molecules, like here you have the amino acids, then the sugars, then small proteins, this can pass to the inside of the membrane via your facilitated diffusion, and this process does not require any energy, and this is happening in a very naturally occurring manner, so it does not require any energy, and therefore it's absolutely working fine. Okay, so what molecules actually pass through that is you have the amino acids, sugars, and small protein molecules. Now you have heard about osmosis. So it is also basically the diffusion of water through a semi-permeable membrane. So semi-permeable membrane is highly required to actually distinguish between the higher concentration portion and lower concentration portion. Now water moves from its own high concentration to its low concentration. Okay, so and then an equilibrium is reached, and after that the process gets halted. So here you see that what is happening over here. So you see that the water will move from a high to a low concentration across the semi-permeable membrane. The last type of transport is basically your active transport. So the movement against, it is basically the movement against the concentration gradient. So this requires energy or ATP molecules to carry out these different transport mechanisms, and it allows for stockpiling, and it's a one direction mechanism. So here you see you have the proton pump, or the what we had studied is a uniport. Okay, so it's an uniport. Okay, so it's a one directional motion. Okay, so you have the types, like you have the proton pumps, and for bulk transport you have endocytosis and exocytosis, one for going out and one for going in. Okay, so this is entirely the active transport, and these are all your basically membrane proteins. So what we need to keep in mind when we are studying about membrane protein is about the different categories of membrane proteins that actually exists in nature. Then we will talked about the different functions which these membrane proteins have, and the different challenges which these membrane proteins face in the scientific world or in the research field. That is the purification step, because the first thing is that it does not overexpress. Overexpression of these membrane proteins are very difficult. The second thing is that during purification you have to purify them using certain detergents. Also the concentration of these detergents have to be you have to be very specific, or very concerned about this particular concentration, such that it does not cross a particular threshold value, or does not cross the a particular line, and you get certain micelles. Although when you see that when you're purifying these against the detergents, at the end of the day you have to remove this detergent, otherwise studying these structure of membrane proteins becomes very critical. Like you cannot perform X-ray crystallography because it would affect the diffraction patterns and all when these detergent molecules are present. And then you cannot also use high concentration of ionic molecules, because it would then unfold your protein, and your protein would be losing its functional or enzymatic activity. And at the end we talked about the different kinds of transport proteins that are present in the cellular membranes. Like you have so in two categories we talked, one is the passive transport and one is active transport. So basically this is the mechanism where you have the osmosis diffusion and facilitated diffusion, and in active transport we spoke about the proton pump and all. So proton pump is basically you take the ATP molecule for opening up the channel, and then your H plus ions pass through it to the inside of the to the outside of the cell. So that is all about the theory portion. Now we would be talking about the you say the practice problems. Okay, so you people would be answering me, and then I would be telling you the answers. Okay, you can give me answers in the chat box. So let us see. So identify the regions marked X and Y. So what is X and what is Y? X is integral and Y is peripheral. Okay, let us see who else is there. You can also answer. Okay, that is the correct answer, because your integral proteins are the membrane proteins that are embedded within the membrane permanently, whereas your peripheral proteins are present on either side of the bilayer, and they adhere with the membranes temporarily and interact with the membranes indirectly by interaction with the different integral proteins or different head groups of the lipid junctions of lipids. So this X is your integral protein and Y is your peripheral protein. That is correct. A is the correct option. Okay, the next one. Yes. Yeah. So which of the following motif is mentioned in the above structure? So what is the motif that we are talking about? So is it an alpha helix? Is it a beta sheet? Is it an alpha helical bundle or a beta helix? What is it? So it's a beta sheet. Okay. Any other answer? Critically look into the picture, you can understand. So what do you see in this particular picture? So these are basically your beta strands, that is for sure. But with these beta strands, you see over here, you have one alpha helix, you have one alpha helix over here, and you have another alpha helix over here. Okay, so there are three alpha helices present. So basically alpha helix, it's not an alpha helix because there are beta sheets. Also it's not a beta sheet because you have alpha helix. Also at the same time it's not a helical bundle, but it's your beta helix because you have the beta sheets as well as your alpha helix. So option D is your correct answer. Okay, because you have a few alpha helices present over here. Anyways, never mind. Okay, the next one. MLFYVW. So these are the amino acids, and this sequence is present in the dash of globular proteins. Interior hydrophilic core, interior hydrophobic core, exterior hydrophobic surface, or exterior hydrophilic surface. Read the question and then answer. So study the nature of the amino acid and then you can understand in which position would it be present. Okay, you're telling B. Okay. Harshraj says B. Okay, can you tell me why do you think that to be the correct option?
So the amino acid sequence we have features methionine, leucine, phenylalanine, tyrosine, valine, which are mostly hydrophobic with only mildly polar properties. And in a globular protein function, the forms a hydrophobic core and a outer hydrophilic surface. So I think it will form an interior hydrophobic core.
Right. On the surface, it cannot be present because then there would be water molecules. Yeah. And the interior hydrophilic core, and so it's an hydrophilic. So these are basically a non-polar amino acids. So it would love to be in the portion where you have a more higher concentration of your hydrophobic residues. Okay, so that is a very good explanation to what you have said. And yes, B is the correct option. It would be present in the interior hydrophobic core portion only. Okay, because all of these amino acids are non-polar in nature. They cannot be present on the surface, they have to be present in the core, but not the hydrophilic, but the hydrophobic core. Okay, good. So choose the incorrect statement about gramocidin A. So what do you know about gramocidin A? The first one, so you have to choose the incorrect statement about gramocidin A. So it works as an ion channel type ionophore. It is a 15 residue linear peptide. It is a ribosomal protein. It consists of alternating D-hydrophobic and L-hydrophilic amino acids. So which option or which options do you think is or are incorrect? Okay. Any other answer? D. Okay. Okay, so let me tell you something about gramocidin A in very brief. So basically, your gramocidin A is a channel forming ionophore. Okay, so that renders biological membranes permeable to specific cations that disrupt cellular ionic homeostasis. And it is also a very well known, you can tell, an antibiotic. However, its potential as a therapeutic agent for cancer has not been studied to a great extent. Now, what happened is that now this gramocidin A treatment is also toxic to cell lines and tumor xenografts derived from different renal cell carcinomas, and it has a devastating disease that is highly resistant to conventional therapies. So, your gramocidin A was found to possess the qualities of both a cytotoxic drug and a targeted angiogenesis inhibitor, and this combination significantly compromised with the growth of different renal cell carcinoma inside your body. Okay, so if you see what the options that are given over here, that it works as an ion channel type ionophore, so that is a correct option. And the next one is, it is a 15 residue linear peptide. It is a ribosomal protein. It is basically a non-ribosomal protein, so option 3 is incorrect. It's a non-ribosomal protein. And then you have is, it consists of alternating dihydrophobic and hydrophilic L-hydrophilic residues. That is also incorrect because it is comprising of alternating D and L hydrophobic amino acids. So there are no hydrophilic amino acids present. So 3 and 4 are the incorrect options, and 1 and 2 are correct. So option D, it would be correct for this one, because 3 and 4 are incorrect. Okay, next one. So which of the following types of ATPase undergoes reversible phosphorylation to transport cations across the membrane? Okay, any answer? No answer. Okay. So let us see. So if you classify different kinds of ATPases, so you can find that they vary or they differ in different categories. Like some differ in functions and some differ in structure. So this category is basically based on structure, where you have F, V, A, and the P type of ATPases. So your F ATPases are basically found in mitochondria, chloroplasts, and bacterial plasma membranes, and are the prime producers of ATP using the proton gradient that is generated by your oxidative phosphorylation in mitochondria, or photosynthesis, that is by the chloroplast. Then you have the V ATPases, that is primarily found in your vacuoles in eukarya, that catalyzes ATP hydrolysis to transport different solutes and lower pH in organelles like proton pump of the lysosome. Whereas if you look into ATP, A ATPases that are found in archaea and are extremophilic, found in certain extremophilic bacteria, and these are arranged like your V ATPases or the F ATPases, and have roles in ATP synthesis. And your lastly is your P ATPase, and your PATPases are actually reversibly phosphorylated by your internal phosphorylation during your transport cycle of cations. So for reversible phosphorylation, you have the P type of ATPases. The next question, you have a
What does
bacterial,
P stand for?
sorry,
What does P stand for in the reversible
phosphorylation.
phosphorylation type?
Yes.
And F?
F, sorry,
Yeah, what does it
F.
stand for? I forgot.
Okay,
A
so
stand.
this was just categorized on the basis on which they found in different organisms and on the different roles. So just like if ATPases, they have roles in oxidative phosphorylation in mitochondria and photosynthesis in chloroplast, and they are found in your mitochondria, chloroplast, bacterial plasma membrane. So they were named. So basically, this F stands for F1, F0.
Right.
Okay, like for V, you have V1, V0. So they named it in this manner, depending on which organism they found and in what functions do they carry out. So on the basis of that. So they basically characteristic on the basis of structure and function, and they named it. So like reversible phosphorylation, you can tell P because it has a similar kind of a abbreviation for it. So like that. Like for AATPs, they found in archaea. So you can tell in that manner. So bacterial porins are beta barrels. Which amino acid would you expect to be particularly abundant at the entrance of a beta barrel that is specific for phosphatidyls? So it should be specific for your phosphatidyls. So your phosphatidyls are negatively charged. So what kind of amino acids do you expect at the entrance of a beta barrel?
Lysine and arginine.
Why do you say so?
Sorry, sir, for I think I answered verbally instead of typing it. And yeah,
No, no,
I
it's
answered
okay. It's okay. You can definitely, because that's
positively
absolutely fine.
charged because of the amine side chain.
Yeah, because of the positively charged amino acids, so they will interact with the phosphate ions, and because of that, the amino acids have to be positively charged to neutralize your phosphate ions or to interact with the phosphate ion. Yes, so lysine and arginine are the correct is the correct option. Yes, that's good. Okay, the next one. All aerobic organisms require dash to produce the basic energy by generating ATP by a ATP synthesizing enzyme called dash through a process called dash transport. So there are three fill in the blanks. Which one is what? Basically, the first one, if I can see, it's oxidative phosphorylation. Okay. So you have to find out an ATP synthesizing enzyme and through a dash transport. Okay, I've got one answer. Yes, that is the correct answer. Oxidative phosphorylation, ATP synthesizing enzyme called ATP synthase, and through a process called electron transport. So that A, option A, is the correct option, right answer. Okay, the next one. Solid band is mostly due to dash transition. It is useful to characterize the dash prosthetic group. So you basically see this solid band in your porphyrin compounds. Okay, so this is the hint that I'm giving you. So you basically see it in porphyrin compounds, and okay, so in porphyrin compounds, what do you have? So you are saying pi to pi star transition and heme group. Any other answer? So in the porphyrin compound, you have a heme group, so that is correct. No other answer? Yes. So option C is the correct answer. The transition is mostly due to your pi to pi star, and the because of the character, it helps to characterize the heme prosthetic group. So option C is the correct answer. Very good. Okay, the next one. Indicate whether the following compounds are likely to cross a membrane by channel or transporter. So one is calcium, water, glycine, and glucose. So would you need a channel or a transporter for passing these molecules? Yes. So what is an ion channel? So basically an ion channel is a pore forming membrane protein that allows your ions to pass through the channel according to the concentration or an electrochemical gradient, whereas your transporter refers to a transmembrane protein that transports your ions small molecules across the cell membrane against a concentration gradient via your active transport. So you would be for you would be requiring a channel for your calcium and water molecules, whereas the transporter would be required for glycine and glucose. So option A is correct. Now the next one. we need to do some calculation. So it says at 37 degrees centigrade, the internal cell concentration of sodium is 20 millimolar and the external concentration is 340 millimolar. Now calculate the minimum membrane potential required to transport sodium out of the cell. So how would you do it, or how would you solve it? So your change in delta G is equal to your 0. That is what I can tell you. Now how are you going to do it? So your change in G, or your delta G, let's write here delta G. So your delta G is 0. Now how are you going to do it? So delta E equals to your,
So can we apply RT upon ZF log outside and concentration upon inside as well?
yeah, that is what is the equation you have to actually apply. Okay, so you have RT, so you have actually minus RT, then you have ln, then you have the sodium concentration that is outside, Na outside divided by Na inside, and that entire thing you have to divide it by Z and F. Okay, so you have a minus sign over here. So I'm just increasing it. So this is the formula. Now calculate and tell me what would be the answer. So the answer would be in minus or plus. So what is the answer that you are getting? So how much is it? 8.314 into 300. Right, yes, into 300, then you have ln of, so internal cell it's given 20 and external it's given 340, so 340 by 20, so ln of 170, and then divided by, so you have over here sodium, so that is 1, the charge is 1, and F is 96500. How much is it coming? Anyways, you just calculate, and here I guess your answer would be 75.7 millivolts. Okay, so your option A would be correct. Okay, just check it once. So you would be seeing that option A would be correct. Use this formula and try to do it, and you would be getting the answer in negative. Okay, just check it and confirm. Anyways, okay, moving on to the next question. If a substance is uncharged, what information would you need to predict whether the transport will be thermodynamically favorable? So if a substance is uncharged, how can you tell about whether um, the transport will occur or not? Means it's favorable or not. Information about pH gradient, membrane potential, solubility of the substance in nonpolar core of the membrane, or information about the concentration gradient. Okay. Yes. So when we are talking about the feasibility or the favorability of a thermodynamic reaction, so for that delta G has to be negative. Now what is delta G? Delta G equals to your RTLN, yeah, the concentration of the ion inside divided by the concentration of the ion outside, and plus you have that particular what to tell, the particular terms where you have the charge with it. Now just because it is uncharged, so that portion becomes zero, and directly it's depending on the concentration of the ions inside and outside the cell. So therefore the concentration gradient becomes very very critical over there. So therefore the information about the concentration gradient is important for understanding the thermodynamic favor, whether the transport will be thermodynamically favorable or not. The next one, the transfer of ions, electrons from NADH to coenzyme Q is hindered by X when it binds to iron sulfur proteins in NADH-Q reductase. Now, blocking the electron transport, Y inhibits the activity of cytochrome oxidase. Z binds to the alpha subunit of NAK plus protein ATPase, and inhibits its activity. So you have to identify X, Y, and Z. Okay, so let us see. What would be the answer? So the first thing it is saying that the transfer of ions from NADH to coenzyme Q is hindered by X when it binds to iron sulfur proteins. So this is actually played by your rotinone. Okay, so X would be a rotinone that plays this particular function. Now you have different molecules that actually inhibits your cytochrome oxidase by blocking the electron transport. You have cyanides, you have azides, you have hydrogen sulfides, and even carbon monoxides. So these molecules inhibit your cytochrome oxidase, and finally your cardiotonic steroids are binds to the alpha subunit of this ATPase and it inhibits its activity. So rotinone, carbon monoxide, and cardiotonic steroids are correct. So B would be the right answer over here. Just keep this thing in mind that rotinone is hindering your NADH2 coenzyme activity by binding to the iron sulfur. The next one. Yes. So which is a isotonic, which one is hypotonic, and which one is hypertonic solution in this following image? So which one is what? Identify ABC. So your hypo, oh, okay, let me see what you're answering. Okay. C, okay. So a hypotonic solution is one that has a lower solute concentration than the solvent concentration. Okay, the hypertonic solution has high solute concentration than the solvent, and your isotonic solution is the solution that contains the same concentration of solute and the solvent. Okay, so what is over here? So water is going out, so that is your hypertonic. Yes, hypertonic. Okay, so we have hypertonic. In B, water is coming inside, so that is your hypotonic. Yes, and C is your isotonic. Yes, correct. So C is the correct answer. Right. And last question for today. Which of the following statement or statements is or are indicative of passive transport? ATP is required for transport. Small organic molecules will diffuse based on molecular weight and solubility in lipids. Molecules are transported from low concentration to high concentration, and molecules are transported from high concentration to low concentration. So which options are correct? Tells. Okay, you're telling C. Any other answer? So the first option that it says is ATP is required for transport. No, this is incorrect because we do not require ATP. So 1 and 4, so A and B are gone. Now you have 2, 4 or 2, 3. So 2 is correct because it will diffuse based on molecular weight and solubility in the lipids. That is correct option. Now molecules are transported from low concentration to high concentration. That is not correct. But molecules will be transported from high concentration to low concentration. So 2 and 4 are the correct options. So here you have C. So C is correct. Okay, good. So that's all from my side. Now if you have any questions you can ask me, else that's all for today then.
Sir, I had a question regarding the quizzes.
Yeah.
The quizzes and assignments you're taking, and it's automatically getting reviewed. I checked some of the answers, and sometimes they don't really match. For example, there was an enzyme-based answer. It was based on if an enzyme coordinates with a metal ion with its cofactor, what does it form? So the options were apoenzyme, holoenzyme वगैरह उसमें तो I chose apoenzyme holoenzyme. That should have been the answer. But the correct answer it was showing in that was enzyme substrate complex, which doesn't make sense.
Okay. But I need to. So it is the question from the quizzes that I taught, or that was in the assignment.
In the assignment.
Okay, then what you can do, you can just means mail me your mail me that question. Okay, I will go through that and tell you.
Yeah, there were actually bunch of questions, like one or two there. There's like sometimes I think I might be making a mistake, but I just because
Okay.
I even conferred with few seniors if they can see, but yeah, they were saying the same thing.
Okay, so in that case, you just put a mail and ask me. I will just answer you because might be the question is asking something different, and there might be sometimes that there might be a problem and all. So that can also be the case. Okay,
All right,
so
sir.
whatever you're telling is right, but you just take a screenshot of that question and just mail me. I will be answering
Okay,
you. Okay.
I'll mail you.
Yeah. So here is my mail ID. You can just mail over here.
Yes, I've copied it. And sir, the lecture, what is it, the doubt clearing sessions? Do they happen like every week around
Yes,
Friday?
yes. They happen on every week from 6 to 8.
Around Friday, no?
Yes, yes. Friday 6 to 8.
All right.
So you people do not get mails from NPTL.
Receive the mails, but sometimes we don't get time in the Saturday
Okay, okay,
batch
okay, okay. But these are
morning.
recorded. These are basically recorded lectures, so you can just check.
I can
Okay.
access it later too, right?
Yes, yes, you can access in YouTube. Yes, you can search with my name only, and you will be getting it. So it's under
All right.
fundamentals of protein chemistry only. I am pasting. Okay, and also I will be sharing the slides. You can take help, and if anytime, any place you face a difficulty, just take a screenshot of that portion and ask me what question you have. I will be answering you over the mail only.
All right, sir.
Yeah. Anything else from anyone? No. Okay. Then thank you all. We will meet in the next week. Okay.